Go back to this. So this is kind of where I got cut off. So here's an example, though, of allosteric regulation. So allosteric, shape change, and it's a change other than a location of the active site. And this is the way we can also look at allosteric regulation. So here is an inactive enzyme, and here's its regulatory site. And so there's two ways that allosteric regulation can occur here. Well, let's take a look. One is that an activator opens up. So actually here's the active site, sorry about that, and here's the regulatory site. So the activator actually enters that regulatory site, and by doing that it opens this enzyme's active site. So now it can do its job. So here's the substrate, and then we see the product. Okay, good. Option 2, we have this inactive enzyme, and instead what happens is an activator, in this case a phosphate group, is going to be added to that inactive enzyme, and in doing so, it's going to open up the active site. So that this is called phosphorylation. So we phosphorylated this inactive enzyme. This particular one is called a kinase, and we're going to see this again later on, but you see the ASE ending on it, so that's helpful to see it. One of the most prevalent enzymes in all of our metabolic processes are kinases, and we're going to see a lot of that. But at any rate, so inactive enzyme, phosphate group, that phosphorylates it, opens up the active site, substrate can come in, do its thing, and then ultimately dephosphorylation would have to take place in order to inactivate this enzyme again. So that's the story. Protein kinases, like I said, are really, really huge in biology. We'll see it in some of the metabolic pathways we're about to get into. And that's basically a lot of what happens with them. They can be inactivated or they can be activated, and the activation can take place either with something called an activator, or it could be by putting a phosphate on it and phosphorylating that enzyme. So these are different ways that can happen. Metabolic pathways that we're going to see here are important to understand. So when you have the very first reaction in a pathway, and I've drawn these on the board where like A goes to B, and then B goes to C, and then C goes to D, etc. The first reaction is called the commitment step, or the committed step. And then once that's happened, other reactions are going to happen following that until you get to the end, until you build up a product. And that end product sometimes can be the thing that shuts off the pathway. So let's take a look. Here's an example where here's our threonine, and threonine is getting converted into alpha ketobutyrate, which is this here, which is like the next step in this pathway. And there's an enzyme right here involved with that. So it's an enzyme specifically for catalyzing this reaction. Notice that after it there's arrow, 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 arrow. Each one of these is saying, okay, we're going to take this and produce something else, new enzyme. Produce something else, new enzyme. Produce something else, new enzyme. So each one of these steps has its own enzyme involved until we eventually get to this end product called isoleucine. This can happen, and you can keep going and keep going, and then you build up this product, this isoleucine, and let's say we don't want to make any more because, the body is pretty efficient. It's not going to make more than it needs. So the end product of this pathway comes back, and it acts as an inhibitor, an allosteric inhibitor of this very first enzyme. This is called feedback inhibition. Some other ways that metabolism can be regulated. So if you get anything out of this course, one thing you're going to learn is that the human body is all about how do we control, how do we regulate these various processes in the way that maintains homeostasis, the best steady state for us. So here are a couple other ways regulation can take place. This isn't always a positive regulation, it sort of depends, but pH can affect enzymatic activity. So sometimes an acidic environment or a basic environment will either facilitate enzymatic activity or could inhibit it. Here's an example. Glutamic acid is going to get converted to glutamic acid here, as you see here. Actually, this is showing the release of this proton, this hydrogen. So what we're doing in this case, glutamic acid is creating more of an acidic environment in one case, but notice the arrow goes both ways. So glutamic acid is basically a buffer that can then pick up extra hydrogen if it needs to, extra protons, and return to this state. We'll look at some specific examples of this, but this is a specific example, but we'll explain where that might be useful. The other regulatory mechanism is something called the law of mass action. The law of mass action is basically talking about, again, sort of with this process you see up here, when you have a high pH, in other words, you have a lot of protons. I don't mean high pH, I mean you have a lot of hydrogen ions released. Remember, that actually would go toward creating a more lower pH, right? This is more acidic. So the higher the proton or the hydrogen ion concentration, the more the reaction is going to be driven to the left. So this is what we were using just a minute ago to say, all right, if we have a lot of hydrogen ions in concentration in the fluids of the body somewhere, then this glutamic acid can pick up these hydrogen ions, and it's going to pull the reaction back over to the environment so that we have more. We're getting the hydrogen ions out of solution. So we're going to the less hydrophilic form when we go this direction. So we'll get into, again, some real specifics on this, but this is just to show you that, again, regulation is what it's all about. And how that regulation takes place, it depends on the situation, but there's a variety of ways. And this is one way that regulation can take place, by changing the pH of that environment when you have a high number of hydrogen, in other words, a more acidic pH. That could cause a change in the enzyme shape, or maybe so we're going to move it back this direction. So we're going to see a change in the ability of the enzyme to do its job, and this is going to control it. So this is regulation. In most cases, we said that proteins, when they hit the tertiary structure, are functional proteins, right? Not secondary, not primary, but tertiary at least, and then sometimes they go to quaternary. But the protein in tertiary structure would be very sensitive to pH changes. Okay, it has it's already got a lot of bonds that are there, right? Protein tertiary structure. Remember, enzymes are proteins. So if you change the pH, you're going to change some of those bonds that occur, or those linkages, like disulfide bridges, hydrophobic interactions, some of the ionic interactions, and any hydrogen bonds can be all affected by pH. Now this is great, and this goes back to your POGL too, so you remember seeing that although they didn't use these particular enzymes, I think, well, actually pepsin was used, I think, but here you have a couple of others, chymotryptin and arginase. These are all enzymes that you see here, and what it's really basically saying is they have an optimal pH range in which they operate. So peptin works really well in the stomach because the pH of the stomach is very acidic. Or these two are more in the intestines, so they're more in this range of operation, where chymotrypsin is over here, so it's a little bit more basic than what we see, this neutral 7 pH, so it slides over here in the basic range. And then arginase is even farther over. Temperature is going to affect enzyme activity too. So we talked about what happens if you have a fever. What could that potentially do? And warming is going to cause a couple things. One thing is it just speeds up reactions. get more collisions. But if the temperature is too high, it could cause some of these bonds to break, like hydrogen bonds. Here's the graph here of temperature, reaction rate. There's an optimal temperature for each enzyme to do its job. Now, we didn't talk at all about isozymes, and I'm going to kind of push that one off a little bit until later. But it kind of bring in an interesting question because it talks about what aspirin does. And aspirin, for a long time, it was a mystery. What does aspirin do to actually take pain away and to reduce some of the inflammation? And what it does is aspirin binds here to this cyclooxygenase, which is something that's involved with our inflammatory response. So something happens, we produce these different types of regulatory products, such as these prostaglandins, which are involved with inflammation and pain. And then the thromboaxines, which I'm probably mispronouncing on that, which has to do with blood clotting. So these are processes the body is doing in response to some injury or insult that your tissue might have had. So even though you say, well, pain doesn't sound desirable, and part of that is because the pain comes from having the inflammation and getting swelling and fluids in an area, and that pain isn't what we want. However, but what we're doing here with aspirin is we're inhibiting this metabolic pathway that would normally occur, and so pain gets taken away. There you go. Here you can see the mechanism of action of this aspirin as an inhibitor, and we're not going to get into the weeds on this one. And it talks a little bit, though, here about how aspirin binds at the active site of this cyclooxygenase and will actually transfer what's called an acetyl group to a serine residue. So what that means really is that serine, so acetyl group it's two carbons and some hydrogen around it. So by binding to this, it's going to inhibit it. And it kind of targets serine. Remember, serine's an amino acid, and that causes the whole thing to change shape, and then that enzyme is kind of inoperative for a while. Here you go. There's the acetyl group. Here's the two carbons. You can see it there. There's some hydrogens, and that's acetyl group. Some of you may remember I talked about methyl groups, and that would be just one carbon and surrounded by hydrogens. In an acetyl group, we have two carbons. If we had a propyl group, we'd have three carbons with hydrogens around. So those terms have to do with hydrocarbons. Here you can see it there, how it modifies that enzyme, this cyclooxygenase enzyme. And there you have it, and that's it. So hopefully that helps, and I will answer questions in class.